if you would please grab your seats because you know we are going to keep this thing on schedule today. Okay, we're going to close down the poster session now, and we have a prize winner, and it is David James. Congratulations. His poster is on zebrafish, and he's exploring kind of GI problems in the zebrafish. So congratulations, David. We will be sending you a check for your prize. And thank you to all of our poster presenters. Okay, if you guys could kind of quiet down now. We need to get started so that we stay on schedule. Our first presenter is Pilar Magoulas. She is a genetic counselor at Texas Children's Hospital, and she's an assistant professor at Baylor College of Medicine. Pilar, you want to come on up? Okay, everybody, please be seated.
Do I need the microphone? Okay. All right. All right. Hi, everyone. Thank you so much for having me. I want to thank Geraldine and the organizers for inviting me and asking me to present today. I'm going to be talking about genetics bright and early in the morning, so I put an exclamation point up there, so you should be excited. All right. This is probably going to be a review for a lot of you. It's just some basic information. Hopefully, you've heard some of it before, but if not, hopefully it'll be a good review. So what I'm going to be talking about today in just this brief 20 minutes is chromosomes and genes, and then the genetics of Phelan McDermid syndrome, genetic testing options, and then the genetic counseling issues. I have that at the end, and I probably won't get to that, but those are things that we might discuss either in the panel discussion, or I'm always available for questions afterwards too. So just to start off, this is a picture of our chromosomes. You can see that they're organized by size. We have 23 pairs of chromosomes, with 1 being the largest and 21 and 22 being the smallest. The last pair of chromosomes are the sex chromosomes, so females have 2 X chromosomes, and males have 1 X and 1 Y, and that determines whether you're a boy or a girl. These chromosomes contain all of our genetic information, which are in the form of genes. And so I'm going to see if there's a pointer. I'm going to use this one because it's closer to me. But our chromosomes, when you unravel them, it's the letters of the DNA, and the DNA encodes genes. And these genes are very important because they code for proteins, and the proteins are what's important for just our normal growth and development. So if there's any changes in any of this process, it can affect the proteins and what our genes are supposed to do. I kind of use a lot of analogies in our genetic counseling sessions, and so I compare the genome, which is all of our genetic information, kind of like an encyclopedia. So you have a lot of different volumes of books. 1 chromosome is like 1 volume. A gene is like a sentence in that book. And the letters of the DNA alphabet are just like our letters. So with Phelan McDermid syndrome, we know that it involves chromosome number 22. And most of the time, it is caused by a deletion on the long arm of chromosome 22. So our chromosomes are separated into a top part, which is called the P arm, which stands for petite. Which is the top part here. And then the long part is called the Q arm. And so when we say there's a 22q13.3 deletion, we say 13.3, not 13, because that's how the chromosomes are separated into sections. And so we're referring to chromosome 22, the long arm, Q arm, and then that specific section, 13.3. And so many children with Phelan McDermid syndrome have what's called a terminal deletion. It doesn't mean lethal. Terminal just means to the end of the chromosome, where the very end of it is taken off. And so instead of having 2 copies of chromosome 22 at that particular area, people just have 1 copy. Some patients or some individuals with Phelan McDermid syndrome have what's called an interstitial deletion, and that means that the very end isn't taken off, but the middle section is removed. About 20% of people will have a chromosome rearrangement that involves 22q13.3, either a ring chromosome where the 2 ends are stuck together, or it can involve another chromosome called a translocation. Oftentimes, these changes will result in an individual having 3 copies instead of 2 copies of another chromosome region, and this is called a duplication. So a deletion is when you're missing a piece, a duplication is when you have an extra piece or an extra copy. When we have a translocation that involves the duplication of another chromosome, it can be very difficult to predict how that child might be affected because there are a lot of genes on that other chromosome region, and so it will depend on what the other chromosome is involved, the size of the piece that's duplicated, and the number and the types of genes that are within that particular region. Now, deletions that involve 22q13 can be of very different sizes. On average, they're around 4.5 megabases in size. And so there can be larger deletions that typically will involve more genes, or smaller deletions that will have fewer genes that are missing. And I believe there's a session tomorrow on genotype, phenotype, and so that might talk a little bit more about deletion size, and is there a way to predict how a child might do based on their deletion size alone. In general, though, we typically see more medical or developmental concerns in children with larger deletions. However, even in individuals that have small deletions, we can see similar features to those with larger deletions. The reason is that there are many, many genes in this particular region on chromosome 22q13, up to 108 genes or so. And there seems to be a minimum critical region that must be deleted in order for us to see the most common features associated with Phelan McDermid syndrome, with Shank III being the most commonly deleted gene. However, there are some reports that say that kids that have 22q13 deletions that do not include Shank III might have Phelan McDermid syndrome too, and I think that's somewhat up for debate how to classify individuals that have deletions of 22q13 but that do not include the Shank III gene, since that gene seems to be very important for many of the features that we see in kids. So with Shank III, instead of having 2 copies of this gene, people with Phelan McDermid only have 1 copy, and that works correctly. This gene is important for normal neurologic function and connections in the brain. So if it's missing or if it's not working correctly, it will cause problems with the brain connections. And Shank III abnormalities are thought to be cause of many of the neurologic features that we see in Phelan McDermid syndrome, including some of the learning difficulties, intellectual disability, some of the language delay, behavior problems, and also seizure disorders. However, sometimes people with Phelan McDermid syndrome will not have a deletion on 22q13, but when we look closer at that specific Shank III gene, we'll find that there's misspellings in the DNA code. And so these misspellings they used to be called mutations, now we call them variants. And so I wanted to talk a little bit about variants, since some of you might have children that have variants in Shank III. So we all have these variants. It's what makes us unique. Some of the variants or changes are benign and don't really cause any problems with how the gene is supposed to work or function. But some of the changes are more serious, and they can cause problems with what that particular gene is supposed to do. And in the case of Shank III, these changes are important for brain function. So if you have a change in the gene, it's going to affect how the brain works. And these are often called pathogenic variants, and so you might see that term on some of your genetic test results. So I like to use this analogy when trying to describe what variants do. And so this is the original sentence. Pretend this is a gene. All of our genes are made up of 3-letter words. And so the original sentence is, the cat had red fur and ran far. We can understand what that sentence is supposed to say and what it means. If someone has a deletion of 1 letter or an addition of a letter, let's say that this D was removed, that means everything else is shifted over. And it took me a long time to just do this, and I don't even want to admit how long it took. But everything else is shifted over, so you can still see the red fur in there, but everything has been shifted, and so it completely changes the meaning of the sentence to where you can't really even understand what it's supposed to say. And this is essentially what missing a letter in the DNA code can do. Some other changes called missense, where you just change 1 letter. And so in this example, we changed the U to an E. This change is probably not as bad as the deletion because you can still understand the sentence. It's not spelled correctly, but it still says fur. It's just spelled differently. Whereas this missense change, the Matt had red fur and ran far, that one does change the meaning of the sentence. And so this is why it can be really difficult sometimes for genetic labs to interpret variants because they're trying to see how significant is this particular change, and does this change really affect how that gene works. And then some other changes are called nonsense, and these are similar to deletions in terms of their severity. And nonsense means that it just adds a period in the middle of the sentence where there shouldn't be a stop. So the cat had red, and then that's it. It just adds a stop in the middle of the sentence where it doesn't even read the whole thing. And so when we're looking at variants, all of these types of variants have been reported in Phelan McDermid syndrome in the Shank III gene. Typically, we see a lot of the nonsense or these deletion or sometimes duplication where it inserts a letter, sometimes it deletes 4 letters, so everything is shifted over. So those are the most common changes that we see in kids that have Phelan McDermid because of Shank III variants. Does that make sense so far? Okay. So benign versus pathogenic. So oftentimes the lab can predict if these changes are going to be benign or pathogenic if that specific change has been seen before in other children with the condition. And so it's really important to look at other kids that have Phelan McDermid syndrome and what's been published. And if that specific change has been seen before in healthy, unaffected individuals or their parents. And so we have a lot of control or population databases where if we have a missense change, for example, and we don't know if it's a bad change or it's a benign change, we look to see has it been reported in just healthy individuals. And if so, then we're less likely to think that that change is actually causing the condition. And Dr. Holder was on a paper recently that reviewed all of these Shank III variants in a recent cohort, in their cohort, and also in the published literature. And that was very helpful in kind of seeing the features that kids with Shank III variants have compared to kids with the deletion. Sometimes, though, we look at the gene, we read every single letter, and we find a change, but despite our best guess and estimate, we still don't know really what it means. And we see this a lot in genetics. We get variants of unknown significance, and they're as frustrating for us as they are probably for you. But this is if a gene change is found, but we haven't seen it reported before in the literature. It's not really present in population databases. We don't really know how to interpret it. These are called variants of uncertain significance. And sometimes testing other family members can help. And so if we were to have a child with a VUS, and we test the parents and the dad also has it, but he's unaffected, then we are more likely to think that's probably a benign change. Whereas if we test a lot of family members and no one else has that variant, then we might think it might be pathogenic, and we would probably look at it a little bit more closely and wait for additional studies to be done to try to see if this is actually causing the features in the child. And so that's kind of the basics of the genetics. And so now I wanted to go to genetic testing options. And so if we go back to this analogy of the encyclopedia and the volumes and the sentences, there's a different genetic test for each of these levels. And so we can do whole genome sequencing, we can look at the chromosome level, we can look at the actual sentence or the DNA letters. And so all of these tests are different and will give you different information. And so if you've had, let me go to it. So the first one I want to talk about is chromosome analysis. This involves looking at all of the chromosomes under a microscope, and we're looking for large, extra, or missing pieces of chromosome material, either entire chromosome or large chunks of it. This can only detect large chromosome deletions, but will miss smaller deletions. We most commonly use this for diagnosing conditions like Down syndrome, which is trisomy 21, or a completely extra copy of chromosome 21, as you can see in the picture. So it'll show us big things, but it won't really show us the finer detail. The chromosome microarray is a computer technique that analyzes a person's DNA compared to a reference or a sample DNA. And so this is able to detect very small missing or extra pieces of chromosome material. And so we're able to see much more detail than just a routine chromosome study. And so what this does, it takes a sample DNA from a patient, which is the green one, and it takes normal or just from a control population DNA, which is the red, and they put it on this gel, and what they try to see is if the color change. And so if it's normal, meaning there's no copy number loss or gain, so there's 2 copies, it will be yellow. If there's a loss, that means that there's, you can see that, oops, sorry. If there's a loss, that means that there's less sample DNA, so you'll see more of the red DNA, and so it'll show up as red. And if there's a gain, you'll see more of the green or the sample DNA. And so chromosome microarray really is a standard for diagnosing most individuals with Phelan McDermid syndrome, and we'll see a loss on chromosome 22q at that particular area on the long arm. Another option, so if we do chromosomes, we do chromosome microarray, and everything is negative, some people, if they still think that Phelan McDermid syndrome is a consideration, they might want to do just Shank III genetic testing. There are a few labs that are currently offering single gene testing for Shank III. I listed them here. Usually it's around 4 to 5 weeks. However, many more labs include Shank III as part of a panel of genes that can include 10 to hundreds of genes, usually autism-related or intellectual disability panels, where Shank III is just one of many genes. Because oftentimes, when we see kids in genetics, we might not have Phelan McDermid syndrome on the top of we might not have it on the top of our differential. We might think it's a possibility, and so when we're getting insurance pre-approval and everything needs to be pre-authorized now with genetic testing, for us, if we're going to be sending genetic testing, we want to try to capture as much as we can. And so we probably wouldn't do a single gene. We might do a panel that includes Shank III or some more extensive testing. And that more extensive testing is called whole exome sequencing, which looks at the coding parts of our gene. And this is a illustration from University of Washington, where when we think about our DNA, we have the coding parts of our gene, which are called the exons, which are the yellowish bars, and then the introns are the gray part, which are just in between. And usually when our DNA is being processed, the introns are kind of spliced out or taken out, and all the exons go together. And whole exome sequencing is looking at just the exons, whereas whole genome sequencing is looking at every single letter on the exons and the introns. And when you look at this little apple picture, the exome really is a very, very small percentage of our entire genome. And so right now, clinically, we're offering whole exome sequencing, and there are some labs now that are going to whole genome sequencing. And so what whole exome sequencing will detect is any mutations or variants in known disease-causing genes. We have about 20,000 genes, but we only know what about 4,000 to 5,000 of them actually do. And so whole exome sequencing will sequence or read every single gene. The test takes usually around 15 weeks, so 3 to 4 months. And insurance sometimes is getting better about coverage for this test. Self-pay, it's around $7,000 to $9,000, so it's very expensive. And so that's why we kind of reserve this for when we really don't know what's going on, or we want to try to capture as many genes as possible in a child with developmental disability or intellectual disability. We get a lot of variants of uncertain significance, so the interpretation can be quite difficult at times. And about 1% to 2% of patients or individuals will have an incidental finding, meaning that we'll find a change that we were not necessarily looking for or expecting. And this is, some examples are in like hereditary cancer genes or cardiac genes that could potentially impact the parent or that child in the future, but it's not the main reason why we were doing the test. And so this kind of test requires consent and more so than other types of genetic testing. So just a summary for the strategy. Since many of the features of Phelan McDermid are similar to other chromosome disorders, it can be really hard to diagnose without genetic testing. And so if a diagnosis is suspected, we usually will recommend a chromosome microarray first, since this will detect most cases. If that's normal, but we still suspect Phelan McDermid, we can consider Shank III single gene testing, or a genetic panel test that includes Shank III, or whole exome sequencing. I think I'm good. Parental testing might be recommended to help us interpret uncertain results, and so if you've had a blood test yourself, that might be why, is because they found a change that they needed some more information to really help interpret what your child's result mean. This will also provide accurate recurrence risk information. I'm not going to talk too much about recurrence risk, but it's something that I'm happy to address maybe on the panel discussion. A majority of the time, though, parental testing will be normal or negative. And so most of the time, the changes in Phelan McDermid syndrome occur as a result of a de novo or just a spontaneous change that happened for the first time in the child and was not inherited from one of the parents. And so here are the parental testing options. So if the child has a deletion on a microarray, we would recommend either a FISH test, which looks just for that specific deletion, or a CMA on both parents. If the child has an unbalanced chromosome rearrangement, so if there's something unusual, either a ring chromosome or if there's a translocation that involves 22 plus another chromosome, we would recommend a chromosome analysis for the parents, because the parent might carry a balanced rearrangement, and you won't see balance changes on a microarray. You need a chromosome analysis to see that rearrangement. If there's a Shank III mutation or variant of uncertain significance found, then we'll do parental testing looking for just that variant alone. So just to summarize, Phelan McDermid is a genetic condition most often caused by deletion on chromosome 22q13.3. This region contains several important genes, most notably the Shank III gene, and this gene is very important for neurologic function. Most cases are sporadic and not inherited from either parent. Parental testing can be performed to give an accurate recurrence risk information, and I didn't touch on this right now, but prenatal diagnosis is available for Phelan McDermid syndrome and other genetic conditions. And I believe that's all that I have.
Thank you.
Okay. Any questions? Or I guess we'll take questions during the panel discussion. Okay, thank you.
Thank you, Pilar. Thank you, everybody. Good morning. Thanks for coming. We're going to put up the poll everywhere now question. Okay, it is up. So please go ahead and log into your poll everywhere, answer our question, and start your discussions. Thank you. If there's anybody that needs help with poll everywhere, please either let your table discussion leader know or let me know, and we'd be happy to help you get there. Thanks. There are two people that did not show up for the field trip this morning, and we just want to check to see if they might be in this room. The name is Espen Ashim. Espen Ashim. And the other name, Inger Marie Salveson. Okay. For anybody that might be here today for the first time and wasn't here yesterday, please do download Poll Everywhere on your phone so that you can participate in these questions. just to let everybody know, Poll Everywhere has been updated, so please do feel free to upload any of your questions and or upvote or downvote any questions that you see within Poll Everywhere. These will be the questions that our moderators are going to discuss and answer. Thank you.
Hey, everybody, we've got about 2 minutes left, but we need our geneticists to come on up to our genetics panel to come back up to the front now. Okay, Dr. Phelan, we need you at the front. And Dr. Betancourt. Dr. Betancourt is being paged. Dr. Betancourt, you're being paged. Okay, everybody, if we can have your attention, Julie is closing the poll now, and we're going to get started. And I'm very pleased to introduce Dean Hartley from Autism Speaks. He's going to be our moderator.
Thank you, Geraldine. Good morning, everyone. If I can have your attention, thank you. I've never seen so many people excited about genetics. This is amazing to me. I mean, most people find it so esoteric that they just start falling asleep, so I appreciate. So I'm Dean Hartley from Autism Speaks. I'm the Senior Director of Genomic Discovery and Translational Science. One of the aspects of what Autism Speaks has been doing, you heard something about whole genome sequencing. This is the option to look through all those 3 billion base pairs to find all the things that are going on within your genome. That was started, and Rob Ring was here. I don't know if he's still at the meeting, but he is somebody who's been really important in moving that genetics forward. So one of our goals with all this, you know, it's interesting to know about genetics, but where does it get you? What does it find? What can you learn from this? And that's what we're really trying to find out about this. And this is a very unique group, and one of the reasons Autism Speaks is interested in this is because, again, the number of individuals that have the Shank III mutation basically end up having autism. So for that reason, it's a learning experience for us as well, so I appreciate that. And I really enjoyed the discussions, and Geraldine, I want to thank you for what a great conference you've had. I've learned a lot, and for that reason, something that's really important is having scientists with families. It's a very powerful statement. Everything that I've ever seen, I come from the Alzheimer's field. I was there for 5 years. When we saw movements in the field, it had to do a lot with families being educated and families being energized to particularly talk to your local legislator. How do you see, again, the dollars for these great researchers that are up here to make sure they have their NIH funding? Families can do that. You can see it in the FDA. FDA is basically wanting input. How much risk do families want to take with individuals in a clinical trial? That's a critical piece for families to weigh in and say, you know what, we're interested in that risk, because FDA is all about safety. So the question is a risk-benefit analysis. So I do think having you come together, discuss this, discuss it with the scientists, is a critical way for us going forward. So I appreciate you all being here, and I'm learning a lot, and I'm humbled by some of the discussions I've seen. So let's get on with this. So actually, why don't we just go through, I know you've met a lot of these individuals, but some people are new. So why don't we just go through, introduce yourself, and just tell you a little bit why you're here. Here.
Hello, my name is Catalina Betancourt. I work on the genetics of neurodevelopmental disorders in Paris, in France.
Hello. Hello. Yeah, it's working. My name is Yonghui Jiang. I'm from the Duke University. I see the patient with many of your kids and request all the genetic testing Pilar just mentioned as my clinic. My research is try to ask the question why your kids are missing Shank III or missing the chromosome cause the behavioral clinical problem you have. So we have a little ability to do in the human, so that's what I choose to sort of animal model. So we create a human form of animal model and then asking what happens if you're missing the same Shank III mutation? Can we learn from human? And from there, then we say, okay, whether we can develop a novel treatment based on understanding we call the pathophysiology in the animal model for mice and the monkey and the fish and all other species.
I'm Katie Phelan. I'm involved because I like to meet new families and see my friends from past years, and hopefully I can help you somehow by writing letters or talking to your doctors or giving you information, and that's what I do.
Pilar McCool. I'm a genetic counselor at Texas Children's Hospital in Houston, and I'm involved mostly with another rare syndrome organization called Cardiofaciocutaneous Syndrome, and have been on their board of directors for about 12 years, and I also love going to family conferences and just getting to meet the families and seeing the kids grow every year.
I'm John Burstein. I'm a medical geneticist at Stanford Children's Health out in California, and I'm interested in the genetics of many developmental disorders and have a specific interest in Phelan McDermid syndrome going back to about 2009. Happy to be here this morning.
Great, thank you. San, can you hear me? No? Okay, great. So let's just start with some of the questions, and the first one that's not, okay. Okay, here we go. I like ice cream. So what additional genes beyond Shank III have been linked to PMS symptoms? Who would like to try to first go after that?
There have been no specific genes linked to other symptoms. There are many other genes that are deleted in patients that have big deletions, but the contribution of specific genes to PMS is not known yet. So I always tell families, for sure there are other genes that can contribute to the phenotype, but what we have shown very, very clearly is that the vast majority of features that characterize PMS are due to Shank III.
Anybody else want to comment on that?
I believe there have been a couple of recent articles, like the one that Audrey Therm was the lead author on, that they noted 44 genes in the deleted region that might have specific actions in the brain that could contribute to the phenotype of people. They were not studying Shank III, or they were studying other genes that might contribute to the neurological deficits. And then there was a study by Thomas Bergeron that he studied other genes on chromosome 22 and even genes that were not on chromosome 22 that might interact to cause the effects of Phelan McDermid syndrome. So there are ongoing studies to look at other genes, but certainly Shank III is the most well-studied gene now.
Great. Let's go on to the next one. Can CRISPR potentially edit mutations in Shank III in the future? So probably all of you have seen recently in the news this thing called CRISPR, and it's something that's hit both the public and the science community by storm because of the simplicity in which editing can be done. There are questions really about the specificity of it, what we call on-target editing, what happens at that specific portion versus off-target. But they're making amazing progress because, one, it's a fairly simple system to use and it's fairly inexpensive. So, will we potentially use this in the future for thinking about PMS?
I can say a little about that. Is this, am I on? No. Thank you. So the CRISPR technologies are very powerful, and they're able to make specific changes within genes, perhaps most readily changes of single base pairs or small groups of base pairs, but people have also used them to make larger changes and engineer segments of chromosomes. I think that broadly, some of the challenges facing the use of that type of technology in a disease, in neurodevelopmental disorders across the board, is that it's very easy in a dish to take a cell and change its genome. It's more complicated to do that within a person, although there are strategies for how you do that. Doing it within the brain, I think, is very challenging, and doing it across the brain is very challenging. So the shortest answer to that question is yes, you can make changes in Shank III or many other locations in the genome using this technology. And in thinking about future therapies, it may turn out that people think about ways to change Shank III or fix mutations, or maybe there'll be some other type of modification that could be made to cells' genomes that would be helpful in the context of Phelan McDermid syndrome. So there's many challenges to intervening in neurodevelopmental disorders, and some strategies do seem more straightforward in terms of how you'd implement them as opposed to directly engineering the genome. For example, medicines that someone would take that would go in through the mouth and the digestive systems or intravenously and end up in the brain broadly and diffusely, like many common medications work, as opposed to having a specific genetic therapy.
Okay, let's go on to the next question. My child has a denovo variant. Should I have my other children tested? Maybe the first thing is, again, to maybe explain what de novo variants are versus what we think about other genetic variants.
Okay. Can you hear me? All right. I will hold it right to my mouth. So de novo, again, means that the change occurred for the first time in the child, and if parents were tested in the blood, that it was negative. And so if that's the case, then typically we would not think that other children would be at any increased risk of having a condition. However, there are some cases of mosaicism where there are siblings born to seemingly unaffected parents, and that's because the change might have occurred and just be present in the sperm or the egg cell. And so when we test the blood of the parent, they don't have the change or mutation, but when there is an increased risk of having a child with the condition, that's really hard to test for. And so when we have a child with a mutation or a change, if it's a deletion, then we can feel pretty confident if the parents test negative that the recurrence risk is very low. We never say 0 in genetics because of that risk for mosaicism in the parent. But if it is de novo, then we would say probably a less than 1% chance of having other children affected. And so unless that child had any symptoms, then we would not recommend testing.
I think one of the things that we've found in the autism world, and a lot of times we've had these what we call trios or quartets. It's usually been mother and father. It's been usually an affected sibling or 2 siblings in some ways. And just through looking at those pairings, we could see, again, maybe a mother or father that was affected that passed on one of those genetic mutations, but then there was actually another child affected in that family that was a de novo. It was not. So what it's doing is teaching us a lot by seeing these genetic pedigrees about the genetics of these disorders, and for that reason it's helpful for us to understand the underlying biology and how we can actually change that. So it's really an important part for us discussing that genetics.
I want to also add, it's also the evolving. We learn more and more over the time, but just 2 weeks ago, there's a new study published on the New England Journal of Medicine. They study for the very severe epi. Hello? Better? Oh, okay. So it should be very, very close to my mouth. So that's true. So that's very severe epilepsy. So before this paper, we all say it's a de novo and the recurrent risk is very low. But this study shows that actually in the germline mosaicism, it's much higher than we initially sort of estimated. So that totally changed how we kind of counsel the family for the recurrent risk. That part is clear, like Pilar mentioned, is we cannot testing for, but then the knowledge we continue still learning more. For Shank III, we don't have that knowledge yet. So, but for epilepsy, very severe epilepsy, this study showed us that's a much more complex question may show up at next meeting or a couple years later.
Great. You guys ask great questions. I mean, they're at the fundamentals of really trying to understand really what the genetics means. So here's another one that I think. How do I find out what my child's specific deletion could mean for their future?
The answer is, if you want to know what the specific size of deletion in your child means, we need you to participate in the registry and contribute your genetic data and your clinical data. You have a powerful, very powerful information in your hands, and we need you to share them with us so we can analyze it and give it back to you and answer this question. PMS, as you know, has very different size of deletions, has mutations of different size, so this is what we call highly heterogeneous. So we need a lot of clinical data to analyze it and be able to tell you if your child has an 8 megabase deletion, he's at risk for this and this and this. If it's a 4 megabase deletion, the risks are this and this and this. Right now, we are unable to tell you that because we are lacking data, okay? You have to understand that in the registry, we only have data with clinical data and genetic information for 300 families. We know that there are thousands of families in the world, but we only have data to analyze in 300 families. And that number of families is not enough to answer that question. So please, please, there is a registry. The foundation has invested a lot of effort and money in creating that registry, but we need the families to take some time, and we know you're very busy and but it's very important. Submit your genetic reports and take your time to answer the questionnaires so we can, in future meetings and in future publications, answer that question.
And Catalina, you'll be presenting some of it tomorrow, right?
Our next question, does 23andMe or Ancestry.com panels cover Shank III? Does anyone know that?
Certainly not.
All right, we'll move on with that answer. So what is the prenatal test for PMS?
So there are a few different options. There's invasive testing, which can be done either with a CVS, which is in the first trimester, or an amniocentesis, which is in the second trimester. And so those are the most diagnostic tests. And so they will take some fluid or samples of placenta from the developing baby. And the test that you would want to get is a chromosome with a chromosome microarray, since again the chromosome study alone might not detect smaller deletions, and so you'd want to make sure that a chromosome microarray is done if your child has a deletion. If there's a mutation in Shank III, then you would want to get actual Shank III testing and looking for that specific mutation, and that's called known familial mutation, and that's usually much cheaper than sequencing or reading the entire gene. There's a new test now called NIPT, which is non-invasive prenatal testing, which is done usually in the first trimester, and this is a blood test that's done on the mom. Usually it's a screening test, so it's not diagnostic. It won't give you a yes or no answer, but this is a screening test for common trisomies such as Down syndrome, trisomy 13 and trisomy 18. There's a newer version that does include microdeletion and duplication disorders, and so it can screen for DeGeorge syndrome, which is 22q11.2, so further up. But most of those do not. If the deletion is smaller than 7 megabases in size, it will not detect the PMS deletion, and so that's probably not the best test for screening for Phelan McDermid prenatally. So it would probably be the CVS or the amniocentesis.
And in the old days, amniocentesis was the non-invasive test, and CVS was the invasive test. But now that there's a blood screen, suddenly amnio's become an invasive test. So I don't know. It's all relative.
So I know we have limited time, and just as a moderator, and to key off of what we just talked about, I want to go to this one question here. When to repeat testing as technology develops? Because we know it's rapidly changing.
I told them they needed to come to this meeting every 2 years and learn what the new technology is. That was the short answer.
For families that were diagnosed a while ago, they were usually diagnosed with karyotype, so very big deletions, or with FISH. None of those techniques tell us the size of the deletion and the details, so I would recommend that for families that were diagnosed with karyotype or FISH, they would need a microarray to determine the exact size of the deletion, okay? And it's not a new technology, but those that are now being diagnosed with microarray, that it's, let's say, the modern technology, every one of you has to understand that in Phelan McDermid syndrome, a microarray is not enough testing. The microarray shows that there is a deletion, but you need karyotype to tell whether that deletion is associated with a ring chromosome. Okay, this is essential. There are 11% of patients with PMS, according to the registry data, that have an associated ring chromosome. Ring chromosome carries increased risk for neurofibromatosis too. So every one of you that has a diagnosis made by an array needs to have karyotype testing. The children, need to be tested with karyotype to tell if they have a ring chromosome.
I had a pleasure kind of interact with a few families yesterday as a part of a consultation. But I think it's a kind of adding this comment, it's if you kids with PMS or Shank III mutation, but somehow your kids' clinical phenotype is well severe than all the other family reporting or share each other. So you might talk to your physician looking for whether your kids may have a second condition or have not looking carefully. So I think that would be my sort of comment. So particular for you kids to say, okay, that's my kids' presentation is out of very, very severe end, me suggesting a second etiology in addition to the PMS or Shank III. We now know two rare conditions come together. It's not so rare anymore from the study we have done last 5-10 years.
I'm afraid our time's up, and there's a lot of excellent questions here, so I think a lot of us will continue to look at them. But I want to thank all the families that have donated their DNA for trying to find out about this and why those registries are so critical for us going forward. So I appreciate all that. And I want to say thank you to the panel for coming up and taking these questions.
Thank you. Thank you, everybody. Coming up next, I'm pleased to introduce our next speaker who will talk about GI dysfunction, Dr. Kent Williams. He is a pediatric gastroenterologist at Nationwide Children's. He has expertise in treating GI disorders in children with autism and other developmental disorders. He's part of a national work group on the development of treatment guidelines for children with autism. He also conducts research on the mechanisms that cause functional GI issues. And I think Ricardo has an announcement to make as well.
So I wanted to make a quick announcement. If for whatever reason you can't be in this room and you still want to see what's being presented, we have a room at the other end of the hotel called Jenkins, where we are live streaming. So for whatever reason you can't be here and you still want to watch it, you can go to the live stream room, Jenkins.
Without further ado, here is Dr. Kent Williams.
All right. Thank you. I have to say, as soon as I came in this morning, I knew I was at the right conference for me. When I walk up and there's a table recruiting poop specimens. Dr. Luna and I worked together, and I saw her name. No, I was just expecting to see a picture of her right by it, you know. Give me your poop. Hands out. So yeah, I treat children with autism. I actually have about 4 or 5 patients with Phelan McDermid syndrome also in the clinic. So, but today what I'm going to go over just briefly, and I have to apologize. This may be a little disorganized. When I looked at the schedule, it was for an hour. I'm thinking, oh, they're going to give me a full hour to talk about things. It's 20 minutes, okay. I just made some edits just now, so we'll see how it goes. So I'm going to go over the most common GI disorders that I see in my clinic. We have about 1,000 patients. Those being constipation, vomiting, which I think has unique concerns within Phelan McDermid syndrome. And then we'll end talking a little bit about the microbiome. So what GI symptoms occur? Well, in our clinic, like I said, I've been there at Nationwide now for 7 years. I did a 5-year chart review just recently for about 720 patients. And what we found was, by far in our autistic clinic population, constipation is the most common thing that we see referred to our clinic. Of the children I saw with Phelan McDermid also, the majority of them, the 3 out of the 5 had constipation issues. One of them, very, very severe, which I'll kind of talk about as we go on. Then the other things are reflux or vomiting. Sometimes it can be a little difficult to determine which is happening, but vomiting seems to be associated with the genetic syndrome. And then even though pain may be a little bit less in this patient population, I do think GI pain is very difficult to determine. And we'll talk about some of the areas of research that Dr. Luna and I are involved in, of maybe actually trying to help identify those kids who are at increased risk for having GI discomfort and GI dysfunction that you're not aware of. First, again, as constipation is the most common thing, I just want to start with a few constipation myths. First of all, having a bowel movement every day does not mean you're not constipated. I get this told all the time when I have patients come to me that the child is having issues, and I ask a few questions. I think they're constipated, but doctor, he has 2-3 bowel movements a day. Well, you can have 5-6 bowel movements a day, and you can be severely constipated. In fact, the more bowel movements you have in a day, the more likely I think you are constipated, and we'll go over that in a little bit. But he's never had a formed stool for the last 6 months, or she's never had a formed stool for the last year. Again, actually, I probably get about a patient every 2 weeks, 4 weeks that comes in for chronic diarrhea, and it's actually severe constipation. And then the other question, of course, then when I start treating it, people always went, well, I don't want my child to become dependent upon laxatives. I don't see that. What I see is if you try to avoid treating constipation, you end up having severe side effects in this patient population. That it's not your routine constipation. It's called mega constipation. In fact, in the autism world, one of the first GI disorders really talked about in the literature was what's called megarectum, where the colon is so dilated on the x-rays, it's mega dilated. These kids tend to have significant complications. They have problems sleeping. They have problems concentrating. They have behavioral issues. Behavior issues sometimes can be contributing to the constipation, but it's not uncommon for parents to tell me, well, I know he must be getting backed up because his behaviors has just been really bad lately. And half the time we look, they're right. Toilet training and social development then, of course, becomes an issue. If you don't treat the constipation appropriately, you're not going to be able to get toilet trained. And then the long-term effects is I see kids probably 2-3 a month where they've had such severe constipation. They didn't start with colon problems, but their constipation is so bad, it's resulted in colonic problems. And this would be the example of one. So this is an x-ray of a young man that came to my clinic who the mom said, oh, we just switched to milk of magnesia. He's having 2-3 stools a day. His behaviors are better. He's doing great. And what we have here is this here is his rectum. That is basically going from his lower pelvis up to his diaphragm. That description there where the radiologist was so nice to say how wide it is, that's 10 centimeters. That's roughly about that wide apart. And this young man, needless to say, had significant problems, but fortunately we've been able to help him. But this is what I'm talking about. This is a family and a doctor came to me, and I have to be honest, I'll be full disclosure here. First time I met him, I missed it too. Then he comes back 6 months later, and I'm like, you know what? Let's just get an x-ray, because things just don't seem to be progressing as well as it was, and here it is. So, was having daily stools, was having soft stools, but yet this is what he was experiencing. So needless to say, discomfort like this, I was having a little discomfort when I saw it. It gave me a little bit of stomach pains. So the criteria for diagnosis, so what is constipation then? The criteria based for constipation is consistency, size, whether you're having to have effort or you have pain with your bowel movements, continence, whether you're able to control it, and then the frequency is really the last thing I ask. Consistency. So we're going to talk about poop. We're going to show images of poop from here on out. Thank goodness thank you. See, I knew this was a conference for me. Walk up. Just be grateful it's not during lunch or right after lunch, is all I got to say. The SHINS conferences I've gone to, they always seem to put me like at 1 o'clock. If you're trying to control appetite, you might want to do what you're doing now. Put me before lunch. All right. So the questions I usually ask are you having hard little balls, rabbit pellets? And believe it or not, there's actually a chart that we use. And if you're really into you can get this right now on your smartphones. Just Google Bristol stool chart, and this will come up. And this is a confirmed way of actually, I can't tell you how many times I pull this up. Parents who don't think their kid is constipated, I show them this, and I say, if your child's stools look like the first two, that is automatically a diagnosis of constipation. Our goals when we're trying to treat, we want nice, soft snakes, soft serve ice cream. That's what I tell people. If you want to see what it's supposed to look like, look up Squatty Potty right now on your phones too. They've got a great video. It's an all-time favorite of mine. Size. The question I always ask, has your child ever passed a stool that amazes you? Are you like, oh my gosh, how did that come out of you? The other one, does your child clog the toilet? Of course, if your child's not toilet trained, there's not really an issue with that. But actually clogging the toilet once a week is a criteria for constipation. And if you say yes, that my child passes these really huge things, automatically constipation also. The effort, pain, does it hurt when the stool comes out? Do they try to avoid it because of pain? Are they straining, holding their breath? Do they sit down and act like they're trying to poop and struggling, but nothing comes out? And how long they stay in the bathroom trying to poop. I mean, if you're staying there 20, 30 minutes and you're actually not on the iPad and playing games, you've got problems. Continence, stool accidents. Now that's an easy one. People will be like, no, my kid doesn't have any problems. He doesn't have any stool accidents. Then I ask the question, are there smears in the underwear? And they'll, yeah, but he's just not wiping well. No. Actually, that is a sign of constipation also. That you just have, your colon is so dilated, just a little bit will keep leaking out. And then the frequency. Frequency, the only time it has a play in it is when you get up to 4 days. When you get to 4 days or longer. So most of the kids that come to me, it's unusual for them to do it. But there are occasionally, of course, kids come in, my kid doesn't have a poop for about once a week. That's automatically constipation too. So Miralax is our go-to standard that everybody uses. I'm going to talk about that in a little bit. But this is constipation that, and the other issue is you got kids with behavioral sensory issues. I would have to say probably only about half the kids in my autistic clinic are on Miralax, not because Miralax doesn't work. A lot of it's because they just don't take it. They don't like it. They can tell the difference in the liquid. The other thing is there actually is a fair number of kids that I give it to, and I can never get them to that soft serve ice cream that I keep talking about. It just is all either really hard or really loose. Other ones that we use, a lot of what I've been going to lately is actually an old standard of mineral oil. I'll have it, particularly the kids that drink milk, I'll put mineral oil in their milk. And this, I think, helps lubricate and actually helps them coordinate and get the stool out. It reduces the sensation. I've been having a lot of luck with that. Lactulose, there are studies showing that it works as well as Miralax. The powder form, actually, it's a sugar, so it tastes a little bit sweet. It dissolves differently, so I get kids maybe to take that a little bit better. There's a liquid form also. Then magnesium hydroxide or milk of magnesia. There's chewable forms, there's liquid forms. Basically, fruit extracts, anything I can get the kids to take. Magnesium citrate, it comes in a powder form now that you can find online. And then colates, docosate, sodium, fiber gummies, fiber supplements, magnesium oxide pills. I will come up with personalized plans with the kids because one may not be the answer or they may not like it or whatever. So I think the problem today, a lot of times in medicine, is if we feel like, oh, Miralax is our best one, let's just use it. And if you can't use it, then I have nothing else for you. Oh, that's not true. We have lots of other things we can do, and I try to be the other thing that we use a lot of in my clinics is stimulants. The two types is bisacodyl and Senna, X-Lax, Senecot tablets. Those, again, people, even the doctors have been told that, I don't want to use that every day. We use it daily. We don't see a problem with dependency. In fact, the reason I think it works in a lot of these kids is it really starts to stimulate. It starts helping them feel the sensation of when they need to go. In fact, some kids, that's all I got them on. I don't even have them on a stool softener. I think one thing, like I said, I give this daily along with it usually. I usually like to make sure the kids' stools are soft before I start giving the stimulants, because if you have a hard ball of poop in there and you start cramping, it can really cause some problems. I have to mention about Miralax. I'm sure a lot of you, and I already heard groans when I said Miralax in the audience. I'm sure there's a lot of people who have seen the multiple news reports that have been in the media over the last 5 or 6 years. There was actually a report out by the FDA that Miralax has been reported to occur with neuropsychiatric events, seizures, tremors, headaches, anxiety, lethargy, aggression, rage, paranoia, mood swings, OCD behaviors. There is that report. We actually were one of the few people that picked up on the FDA said where they had analyzed lots of polyethylene glycol, which is a polymer of ethylene glycol, and what they found is there was trace amounts of ethylene glycol and diethylene glycol in it. We actually did a study. We're the only ones that have published on this aspect of polyethylene glycol. There's been numerous studies of polyethylene glycol and Miralax out there showing its safety and things, but it wasn't looking at the potential toxicity of it. We did do it. What we found was there is trace amounts of ethylene glycol and diethylene glycol in the Miralax, but it's like 0.00001%, extremely low levels. What was more astonishing to me is when we started looking at the children. What you have here is we have a control group of 18 kids, never ever seen Miralax in their life, and we got there we go. We got 9 kids who were chronically on it, a full capful a day between the ages of 7 and 12. What I want to point out to you is everybody had exposure to ethylene glycol. We haven't found a person yet, even in our lab, adults, who doesn't have detectable levels of ethylene glycol or diethylene glycol in their blood. So we're exposed to this constantly in our environment. Now, it's not a naturally occurring. There you go. I'm getting a little schizophrenic myself now. So what this shows is that there is no difference in levels between the two groups who are chronically on it, never seen it. The interesting thing is the level in one child who had never seen Miralax was twice as high as anybody had seen in our Miralax group. And the same goes for our diethylene glycol. It actually was significantly higher in the control group than in the Miralax group. For whatever reason, we don't know if it's just the random 1 in 20, that if you do a statistical study, you have a 1 in 20 chance of being wrong. Maybe this is the 1 in 20 chance. But we found it very interesting, and again, that the highest levels we found were in kids who are not on it. So, and not only that, but the amount that we saw in there are actually standards for having toxic exposure. To get to toxic exposure for a 10-kilogram child, which is a 1-year-old, basically, an average 1-year-old, you would have to take about 34 ounces of water with 50 capfuls of Miralax in a day to even remotely get, and that's for a 10-year-old. Now, older kids, we're talking, a pool of water. Or a liter of water, 15 capfuls per day for 10 days. I have never even remotely come close to that. And that's just to get to the lower limits of concern. So the point being is this causing toxicity from this standpoint. Now, there's other aspects of whether it changes the microbiome and those kind of things, and I think that's legitimate. But that's going to occur with any stool softener. In fact, what I typically see in my clinic, all those reports that went along with it, I see it in kids with constipation. We did a study where we looked at autistic-like behaviors, not trying to necessarily diagnose autism, but autism-like behaviors, which would be the mood swings, the lethargy, the seizures, the tics, all those things that you would typically see. We screened 100 children, and 30% of them screened positive. And we sent those children to go be evaluated, and only 15 went. And out of that 15, 9 children never known to have a behavioral disorder were diagnosed with a behavioral disorder, a genetic syndrome, learning disorder, and other issues. So there is a close association of behavioral issues with constipation. So success or failure, what we typically do is if you're going to end up being treated in my clinic and you have behavioral issues or autism or Phelan McDermid, you will probably be on something a minimum 12 months. Because we got to get your child to be able to have routinely bowel movements and things where it's not a problem. And then we slowly wean off. If you're having problems, we then go into having more aggressive therapies, and we do motility studies. We actually do Botox injections in the anal sphincter to help be able to get the stool out. And then there is a surgical procedure where we actually are able to flush the colon daily with a cecostomy or Malone. But I can talk about those other times. The other thing I want to talk about before my time runs out is vomiting. There are special considerations in Phelan McDermid. There's seizure activities. One of the children that had severe constipation had an x-ray that looked similar to what I just saw. She had severe uncontrolled seizures. When she ended up getting her seizures under control, her constipation got better. There's not a lot of data in the literature, but definitely anecdotally in clinics, if constipation gets worse, seizures get worse. If seizures get worse, constipation gets worse. So there's definitely an association, I believe, between the two. So we have to make sure for that. But for vomiting also, there's been plenty of times where a kid will come to me for vomiting and they're having seizure issues that are not become. So it has to be considered looking for seizures if your child starts vomiting. Arachnoid cysts, increased intracranial pressures are things. And then cyclic vomiting, which is just recurring episodes of attacks of vomiting, which seem to occur more often in these children. So there's a lot of brain-gut interaction. So the last thing I want to talk about is that we did a study with Dr. Luna where we looked at the microbiome in autistic children, and what we found is that children with autism had distinct differences in their microbiome compared to neurotypical children and neurotypical children with GI issues also. And so there appears to possibly be a unique profile. What was more interesting is we actually started seeing a distinction between kids who had abdominal pain and those who didn't, which we're in the midst of doing analysis on the study to see if that can be further evaluated. So looking for a biomarker for abdominal discomfort, abdominal pain in children who can't verbalize well could be extremely useful. Thank you.
Thank you so much. Thank you. So everybody is welcome to go to their app, the Poll Everywhere app. There is a question that is up and ready for you, and then please discuss. Thank you. Sorry to interrupt your discussions, but just to let everybody know, we are now switching the poll everywhere over. So please feel free to upload your questions and or upvote or downvote any questions that might be pertinent to you. So our panel will be happy to answer them in 5 minutes. Thank you. We have about 2 minutes left, and then we're going to start our roundtable. As Dr. Ruth Ann Luna.
So I didn't get to plug you as much as I wanted to, but I still did.
I'm amazed that you got through all of that. you had the crowd rolling. I loved it. You needed that break, I think.
Hi, guys. It's my pleasure to introduce you to our panel today.
Can I get everybody's attention, please?
Hello, hello. Testing, testing.
We have Dr. Billy Bennett. I would like to speak to everybody.
Hi, guys. My name is Billy Bennett, and I'm going to moderate the GI dysfunction panel today. So I have a daughter who has Phelan McDermid. She's 2.5. Her name is Cecilia. If you see a little red-haired girl with a pink dress on and curly hair, who's like really, really cute. I know I'm biased. Then that is her. And then I'm also, coincidentally, I'm also a pediatric gastroenterologist at Indiana University. My clinical focus is on families with special healthcare needs and GI problems. And then I do research in how we can use informatics to improve how doctors deliver care, and then also something called patient engagement, where we figure out ways to better communicate with families. And then on our panel today, and I want to point out that the GI panel is the smallest panel, but we're also the most important panel, obviously. Thank you. Yes, thank you very much. And so you guys have met Dr. Ken Williams from OSU. I'm sorry, The Ohio State University. And then, well, you know, OSU means different things in different parts of the country. You have to be careful, you know. And then Dr. Ruth Ann Luna from Baylor. And I think let's get started with Dr. Luna. This question is tailor-made for you. What about the use of probiotics in this patient population?
Is this thing? Yeah, it is on, I guess. This is the hold it close to your mouth microphone. So the probiotic question, and one thing I'll mention too is that obviously I'm a scientist and a microbiome researcher, and please go visit our table out there. You can't hear anything? Okay. You really do have to put this thing completely on your lips. I feel like I want to sample the microbiome with the microphone after we're done here. But getting back to poop, we do have the table out there that we're actually now recruiting to determine the microbiome that's inherent in Phelan McDermid syndrome. So please go by and see our table. We'd love to have all of your participation, similar to the conversation earlier with the registry. This is considered a poop registry that would be vitally important so that we can answer a question like probiotics. So I do a lot of work in the microbiome of various neurodevelopmental disorders, a lot of work in autism spectrum disorder. We've also recently completed a study in Rett syndrome. I'm also the mom of a 9-year-old son with severe autism, and basically the entire list of GI symptoms that Kent went through earlier that I didn't recognize myself. And this is what I do on a daily basis, right? In probiotics, we don't know for sure. We can't give you a great answer, but we have to, I often say we have to figure out where our starting line is before we can figure out where our finish line is. And we find that starting line by characterizing the microbiome from the stool samples you provide us. If we see something abnormal that we think we can positively benefit by the use of something as easy, relatively easy, as a probiotic, then this is something that we need to test in very carefully controlled trials. And I know Kent and I talk about this routinely, and that, you know, rarely can you do any harm by giving a probiotic, but we already have our plates full as it is, right? We want to make sure that there's a possibility of some great benefit, and we've really got to figure out what that baseline microbiome is before we get too far down that road.
I want to make one comment about probiotics, that currently probiotics are not regulated by the FDA, so whenever you buy probiotics over the counter, they vary widely in price. And all of them say that they are amazing and that they can cure all kinds of things, like your cancer and your ADHD, and that you'll get $1 million the next day. But just recognize that currently, like any advertisements for probiotics or the products that you buy, they aren't required to be tested, and they aren't, and that many times they don't contain what they say they do. And so that's all the more reason, like Dr. Luna says, that we need to study this better so that we know precisely what we need to be giving people, and not let anybody be ripped off by this sort of unregulated products. Dr. Williams, do you have any thought about this?
What I usually talk to families about is that it's, could have some benefit and with low risk. But what I usually tell them is I wouldn't just put a kid on it and just leave them on it. I would try to identify specific behaviors they're looking at, whether it be meltdowns, concentration, sleeping, whatever, and go on it for a month and then go off it for a month and see if you really see a difference on and off. If you don't, then don't spend your money.
So the next question I think is pretty relevant for most families, which is how do you think the high pain tolerance and hypotonia impact how the colon functions and the possibility of constipation? That one's wrapped up for you, Dr. Williams.
Yeah. Can I show my picture again? The x-ray?
I actually tried to go back on the slides yesterday, and I was told emphatically no, that we have to stay.
That's how I think it impacts it. I think the low tone, it's hard for the kids to necessarily because it's not just muscles in the colon, it's your abdominal muscles. And to have a bowel movement takes a lot of coordination. You have to recognize the sensation, you have to process that information, and then you have to try to coordinate all the muscle groups to do that. And the low tone, not only in the colon, but also in your truncal and abdomen, can affect you having constipation. And then the pain perception, I'm still not clear on in this group how that works, but if it is less, then for sure, as you saw in that picture, that wasn't a picture with Phelan McDermid, but I had a child with Phelan McDermid that had an x-ray similar to that. So obviously not, I mean, that would cause pain in most people, but doesn't in some of these kids. So it definitely alters it. And plus, I think the pain perception then comes into how much of the sensory processing is affected. So how much do they actually recognize when they have to go? Would really be the next step. And so in this patient population, those stimulants that I talked about, I would use those fairly frequently in this population because of the low tone and the sensory issue.
Okay, and then here's another sort of fascinating topic that I think both of you could comment on, which is, what do you think the role of diet is in patients with Phelan McDermid? And I think, specifically addressing some common diets which are used, such as a dairy-free diet or a gluten-free diet, and how does that impact symptoms, and then also how may it impact the bacterial population?
I think I can answer that a couple different ways, and one of them is anecdotally. And yes, there are a lot of common diets that are utilized with people saying this is a miracle diet, or I saw no difference at all. And it goes back to tracking data in your own household. If you're going to remove gluten, if you're going to remove dairy, what positive benefits are you hoping to see, and can you take some data to see if you're getting better sleep, if you're getting fewer behavioral issues, if overall they seem to be a little calmer. Those, whatever your problems, your largest problems are at home, see if these dietary changes make a difference. We personally went gluten-free, dairy-free for a certain period of time and immediately saw benefits. Well, if you go back and try to view it objectively, anyone drinking that amount of milk a day would likely be having GI issues. And so it was less about the fact that that specialized diet was hugely beneficial for his condition, and more about the fact that his nutritional choices and maybe my ability to moderate those nutritional choices could have been improved. And so we had this positive benefit, but it wasn't necessarily due to the specialized diet. It was just due to rethinking what it was that he was eating or drinking every day. And I think that's very much the case that you see in many of these diets. Again, it's important to have carefully controlled data as to what's changing if you're going to make those dietary changes, and to make sure that you're not accidentally causing an additional nutritional issue. So always do it under supervision of your GI physician or even a nutritionist that can help guide you for your specific case. And I know Kent said that earlier, that every kid, he will basically follow their lead and prescribe something very specific for them. I think it's very much the case with diet. We're still trying to unwind how the diet impacts the microbiome, and certainly there's a lot of beneficial aspects of that, but again, we're still figuring out where that starting line is. So somebody telling you that we can absolutely change this by removing that, if we don't know that back in the lab, I can't say that somebody out there is going to know that any better than we do. So
yeah, we're a good team. Good start. To expand on that, so when you're identifying the behaviors, I usually also encourage the families, get a third party. Get a teacher, a care provider, another family member who doesn't know what you're doing with the diet, and have them track those behaviors if you can. That would be one way of looking at it. It's the same thing with the use of the probiotics, medications, just to see if there's any kind of change. The interesting thing about the gluten-free, casein-free diet is really the only one that's really been studied as compared to GAPS diet, carbohydrate-specific diet, bone diets, paleo diets and things. But in the gluten-free, casein-free diets that were studied in the autistic population, there's only really 2 studies that came out that were blinded. And in both of those, they didn't find any change or any effect when they tried to validate it by looking at the behaviors with a behavioral analyst. Interestingly, one of those studies, even though they told the families that there was no change, the family still elected to continue the diet once they started it because they thought there was differences. I also agree that I wouldn't do it without supervision. I would even go further, though. Don't go to your GI doctor. Go to the nutritionist. They're the ones that will really be able to help you with it. Because if you're taking out gluten and casein, plus some of the carbohydrate-specific diets and other things, you're removing a lot of sources of calcium, zinc, the vitamin B complexes and things. So make sure you're not missing some minerals and vitamins and those things. Interestingly, in that same study I was talking about, what was interesting to me in one of them was they did what was called a healthy eating index of the families who were on a gluten-free, casein-free diet, which means like in the complexity of the diet and the variety of the diet. And what they found was in the gluten-free, casein-free diet families, their healthy eating index was actually higher. So what I think really the real underlying take-home from special diets is getting rid of processed foods. Getting rid of, potato chips, getting rid of macaroni and cheese only, or only peanut butter and jelly, and really making the kids then eat a wider variety. If you take out gluten, you will take out a lot of processed foods. If you take out dairy, you take out a lot of foods that are high in fat and maybe have high in protein and things like that. So I think a lot of it has to do more with the healthy eating index than the specific diet.
That's a very good point. So one slight modification. So a nutritionist is somebody that just says that they know about nutrition and they hang up a shingle and say, pay me and I'll give you advice. A registered dietitian is someone who very specifically has training, licensure. They take a very difficult test. I actually have taken the test and did not pass, and I'm a pediatric gastroenterologist.
Yeah.
So just so you know.
Well, you prove my point. I mean, that's what I find daily in my clinics is the registered dietitian.
Yeah.
You're exactly right. They do a much better. I gave up after they would come in and start doing it. But that's because we have the ability to have them in our clinics and in our services. But a registered dietitian, I wouldn't do a special restrictive diet without going to one.
Absolutely correct. But and that's something that, and I have an RD that works with us that deals a lot with the special needs kids. And I think it's important to be flexible because people are going to be comfortable with different diets. But if you do it with the help of an RD, then you know that you're getting all of the nutrition you need and the micronutrients that you need. If somebody says they're a nutritionist, anybody could do that. I mean, even I could do that, and you don't want me doing it. But just be aware that that terminology is important, and it indicates licensure and knowledge and that sort of stuff. Okay. I think I have at least like 40 or 50 more minutes. So let's talk. Actually, I want to address one question from above, which was, is standing to poop long-term going to cause a problem? I will just say no, it will not.
Actually, it's more physiologic.
Yeah, it's better. I'm sorry.
It's actually better. Actually, the Western culture has done itself a disservice by making us sit down on a toilet. I mentioned squatty potty earlier. It's actually legit. It puts you in a squatting position. It will put you in a sitting position, but it puts you in the same formation as if you were squatting and standing. It actually is easier to get the stool out because when you sit, the floor of the pelvis comes off and basically closes off the canal, and you have to generate more force. So if you have kids with low tone to begin with, that becomes a problem. So actually, it's fine.
Okay, so then next, there's a lot of questions that have to do with reflux. And so if you guys could comment on the reasons why and potential treatments for reflux, which is a pretty broad question, I realize.
So what exactly was?
So does a PMS, let's go with this. How about, does a PMS child outgrow reflux? Or does a child with hypotonia and special needs with neurological dysfunction, do they outgrow their reflux?
It's possible, yes. But because of the hypotonia, because if they're not ambulatory and they're sitting in a wheelchair most of the time, then they're at increased risk of kind of always having issues with reflux and probably need to be followed. But any child, as they get bigger and as they start to stand and walk more, often will outgrow reflux if they had it in the first year or two of life.
And I would add to that that sometimes it's not just reflux. Sometimes it's the stomach doesn't empty well. Other things that have to do with low tone in the GI tract. So seeing this is something that pediatric gastroenterologists actually are good at, and it's worth exploring. Beyond just giving Zantac, it's worth exploring if there's more going on.
I agree with that too. There was actually the abstract over there talking about the stomach function being altered with Shank III mutation. I don't think we really know at this point, because the same neurons that are in your brain are throughout your gut too, and if there's an instability in the neuronal connections that could possibly affect motility, we just don't know at this point. To go back to his point about there are so many other things. If there's allergies, if there's asthma, skin rashes and things, then you need to be looking for those same kind of things that may be affecting the esophagus also.
Okay, so there's several questions asking about hypotonia in the GI tract, and is there a way to specifically address low tone? And there's lots of treatments for the limbs and the skeletal muscle and things like that, but is there a way to address this tone in a general sense within the GI tract?
The medications that really change tones, particularly were more for motility and things, actually that have been found to be work are no longer available because they had systemic side effects of cardiac strokes and things like that in the States. There's no real approach for increasing tone in the GI tract at this point. What we typically do is try to find things to minimize the effect of low tone, which would be maybe, particularly with reflux, would be maybe smaller, more frequent meals so that the stomach doesn't have to empty such a large amount, doesn't have to handle such a large amount. Same with the colon, to be on a daily bowel regimen so that the stools are soft and easier to get out and don't have a buildup that would stress the system. So really there's nothing specific for addressing the tone at this point. We would use stimulants thinking that might address, but we're not really 100% sure whether that would really increase the tone at this point either.
Yeah, yes, sir.
Yeah. We don't really know at this point. Usually what people are asking me on those questions is whether they're contributing to their constipation, because we know Depakote can alter bowel function and those things, but we don't have any information at this point, or do we?
No, we don't. That's the party line. However, I can tell you that there are a lot of studies in mouse models that are looking at the effect of these drugs that you're mentioning on the GI tract as well, and they're showing that they are increasing. There's a benefit in total GI transit time and colonic motility and
yeah. You bring up an interesting point. I told you earlier that I had one of the kids that came to my clinic with Phelan McDermid that was really severe constipation. And we were getting ready to do surgeries on her for her colon. And she had uncontrolled seizures. And so we worked with the neurosurgeon and neurologist. I said, you know what? I would rather get her seizures under control first before I'd want to do any surgery or do anything. And so they did. They did a severe, not that everybody should get this, but for her it worked, where they did a hemispherectomy where they cut the brain in half. And lo and behold, she was a new person. Her seizures were under control. Her consciousness was more. She was more alert, more interactive. And lo and behold, we went from a kid who was needing to be disimpacted almost on a monthly basis to normal bowel movements. So I'm not sure the lithium and Depakote may necessarily help the gut itself in these situations, but if it helps the awareness and the alertness and the sensory processing centrally, then it would make a huge difference.
And the one thing I'll say there, drugs targeting serotonin and the serotonin pathway, especially, we've seen those really cause a positive benefit, again, in the guts of mice, but certainly on the right path. And anecdotally, we are in our own storm at home right now because we took our son off a psychiatric medication, and we are in the GI disaster phase. Again, that's not something you're ever warned of, but keep track of these things, and you'll see both positive and negative benefits of that.
Thank you very much, Dr. Williams and Dr. Luna, for answering questions.
Hi, how are you? I have a comment just to make. My name is Baha Moshri. I'm a motility specialist, actually. That was our poster that won. David did a great job on the zebrafish model in autism. And just to interject and make a comment, there is a test that you can do to, and it's not invasive. It's a gastric emptying study that's done by liquid emptying, and it tests the emptying of the stomach. And so that's an easy test that usually is not done commonly unless the patient really has severe nausea, vomiting. In the pediatric population and in adults, we do it more commonly, especially in diabetics who have hypotonicity of the stomach, like everyone here was alluding to. So there are some tests, but as they were saying, the FDA-approved medications are actually, there's not that many of them. There are some antibiotics that can help, but these things, again, are, a lot of the medications are purely on research.
Little caveat to that, though. I'm actually one of the PIs of the Gastroparesis Consortium. And the caveat to the gastric emptying test, I use it, don't get me wrong, but all the studies are based upon adult. And the early studies we have right now is that the gastric function may be different in children than in adults and somewhat delayed. So I do use it, but a lot of it is we will also just treat with the same medications regardless and go more on the clinical history. So I use it when, especially if someone was having recurring vomiting, I for sure would be getting that.
Thank you. Actually, we have to stay on schedule, so we're going to keep going. Thank you so much to everybody. Please feel free to ask questions directly of the researchers. They'd love to answer them.
We're gonna try to do a meetup with a couple of the GI doctors after the sessions are over later this afternoon, and see if we can find a meeting room. We'll post on the conference app if we're able to find a location and if the doctors are available, because we've got several questions. Sorry.
Thank you, Veronica. Next up, our next session is going to address a common problem that strikes across all ages in PMS: regression. Dr. John Burstein will tell us about some of his findings from his recent paper and other research. Dr. Burstein is a pediatrician and clinical geneticist at Stanford. His research interests include autism and other developmental disorders. Welcome.
Good morning. I'm happy to be back with everyone here in our community, Phelan McDermid syndrome. And I'm here today speaking, is that better? Just hold it. Okay, that's much better. I'm here today speaking on behalf of my own work, but certainly done in partnership and with leadership from colleagues of mine, Joachim Hallmeyer and Ruth Ahara, who aren't able to join us this year. My story of learning about Phelan McDermid syndrome began around 2006. I was a trainee in clinical genetics. I was at an outreach clinic in rural Northern California, and I saw a toddler with Phelan McDermid syndrome. And my mentor at the time, who had written one of the early reviews describing some aspects of the condition, pointed out that this is sort of, John, what you should understand a child with Phelan McDermid syndrome to be like. And it was a child who wasn't speaking, didn't have language, and the connection I was instructed to make is that children with Phelan McDermid syndrome, they have these large chromosomal deletions, and it's what you should think about if there's this extremely limited communication. Subsequently, I've learned a huge amount of the condition and got involved with research on the condition initially through the effort working with Ricardo Dolmetsch and working with many of you on developing a biobank to study the IPS model of the condition. And as I met more and more families, we all learned, and I learned, that the genetic diversity of the condition is much larger. It's not all deletions, and the range of the characteristics of the children and their trajectories and experiences is very different. I can tell you I was quite surprised when I was greeted and a child with Phelan McDermid syndrome said hello and started asking me questions, because I was told that really wasn't part of it. And as we got involved in the community and had this cohort of patients. We have a cohort now of trainees and investigator scientists at Stanford who have at different times asked, what else can we learn about the condition and what might be interesting. And we talked here a few years ago about work we've done on sleep, and regression also came to our attention. And initially we hadn't, and I think others hadn't paid as much attention to it because it was, well, there is some regression. It's happening in toddlers. It's maybe similar to what happens in autism, that children may gain their first few words and then lose that. And we had, so Jillian Ryerson, the lead author on the paper I'll talk about in a little minute, she got interested in this and she looked at the database of the volunteers, many in the room, families, who had shared their information with the registry and their biosamples. And then thanks also to Sean Berquist, who was a research assistant with us, who traveled across the country completing many of the assessments that the work is based on. She said, well, John, I'm interested in regression because it doesn't seem like a phenomenon that's really only happening in the very youngest children. It's happening in these older children. There's like these 6-year-olds and 12-year-olds and 20-year-olds. And I thought that was really interesting, and other people have certainly noticed this ahead of me as well. And if you actually look back to some of the landmark studies really building the associations between the deletions and the point mutations and Phelan McDermid syndrome, there were patients in these series who were noted to have regression. Some of this just by survey, so there weren't standard criteria and such, but in work that both Phelan and McDermid were involved in, almost a third of the families had reported some regression. When we moved to the discovery of the role of the point mutations a few years later in 2007, actually in 4 of the patients reported there, 2 who were toddlers and 2 who were adolescents, there was also regression. But I at least was not thinking about this that much at the time. Some of the ambiguity about what's been going on here has been that many of these early studies didn't have sort of a formal definition of what's regression or standard measures, and relied on parent report. But parent report is a very powerful thing, I think, as we'll learn together during our discussion today and the discussion that's also going to happen tomorrow. So developmental regression you can think of in terms of words, children making developmental progress, and at some point that decelerates and may even reverse. And you can use standard definitions about the amount of time that a skill has to be present before it disappears. You can also think of this, I'll use this slide. May have been warned not to go backwards, I might do that. But think of a developmental trajectory. No child actually has a developmental trajectory that's as smooth or as linear or nicely curved as these pictures. There's a lot of fluctuation among all children week to week and month to month through periods of different experiences and greater or lesser stress. But the big message about regression as a phenomenon is that there's not continued upward progress. So there's either a plateauing or actually a reversal of skills. So for example, a child may gain words and no longer use them. They may be able to dress themselves, no longer dress themselves. And one of the areas where this has been most studied has been in autism broadly, not just autism in the context of Phelan McDermid syndrome. And it's been found by many individuals that up to a third or so of children with a diagnosis of autism will experience regression at some point in time. And typically this is at this young age, around the time of first words and first steps shortly after that, so between 1 and 2 years of age. That's what was so striking to me when Jillian came to our group and pitched her project about looking at regression in Phelan McDermid syndrome, because it seemed like there were things going on in older children and it wasn't clear what that was all about. So again, we had access to so much of the data that our community has provided, had been gathered by hardworking colleagues of mine, and looked through that. So we looked at the results of the ADI, the Autism Diagnostic Interview, as well as the scales of early learning, and when it was completed, the Stanford Binet. And there were 50 individuals in this cohort, for 42 of which had completed all of the measures relevant to looking at regression. So there are subscales within the ADIR that can be used to get sort of a narrow window onto regression. It's regression in the context of autism, and we'll see why that's a little bit limiting, but that's sort of where we started this portion of our learning. So there were boys and girls present and representing a wide range of ages, which helped us see what was going on in the older kids. And in terms of the a unidimensional measure of functioning in terms of nonverbal IQ, there was quite a range, somewhat representative of what's been seen over the years in children with Phelan McDermid, ranging from moderately high functioning to the lower functioning. So as I was saying, within the ADIR, it's a standardized set of questions that can be administered to a family. Many of you may have undertaken this with a psychologist or a research assistant. And it's defined specifically that if you have a skill for 3 months and then you lose it for a period of 3 months, that can be defined in this context as regression. And it can apply to speech as well as to many other domains. And this is also something that Jillian picked up on early, was that it wasn't all about losing a word or two. There were some things to suggest that this could involve a motor and self-help skills, so things about walk and dressing and other dimensions can be looked at on this scale. And taking all of Jillian's work together, what she saw was pretty different than what's seen in autism broadly, and that's that the skills that were most commonly lost, and this is not 50% of the children are losing this, but amongst the subset that did show regression, which areas were most impacted. So of the 18 individuals who had some evidence of regression through the ADIR, it's actually the motor and self-help skills that were impacted, followed by language, and then the other domains. So this, in terms of the nature of the skills changing, seemed to be very different than from what was understood from autism, and then the ages were different as well. So this is a graph showing age across the bottom, and then age at which there was the onset of the regression symptoms. And you can imagine that if you're only 3 years old, you can't have regression at age 6. So there's a bound in terms of the lower left side of this graph. But you see, I think I have a pointer, I'll use it here. Yeah. So here are children who had regression in toddlerhood, and there's a number of them. And then there are these older children and adolescents going on even to adults who had change in their measured abilities later on. So 10 years, 15 plus years. And that seems different. Even though we're using the word regression and discovering this on the same scale, it begs the question, is this or isn't it the same thing? Jillian also looked to see if there were clear correlates between the size of the lesions that patients had and the frequency of seizures. And in our relatively small data set, there wasn't evidence for these sorts of things. So broadly, it was found that there were 43% of the subjects had experienced some sort of regression as assessed by the ADIR. On average, and the average isn't really necessarily that descriptive here. It was at 6 years of age. That's what drew our attention to it. But perhaps there was, it's still a hypothesis, but maybe a typical presentation of regression in toddlerhood that looks more like what people describe in autism, and then other things happening to people when they're older. So in preparing to come here today, I learned some more things, looking very thoroughly through the literature helped out by some well-written reviews and some of our own work. And it's, there's been clues and bits and pieces about regression and Phelan McDermid syndrome in the literature for a long time, noticed by a lot of people, and some have done good work in pulling it together, and I hope I can add a little bit of that this morning and foster a good discussion. So to me, they seem to fall into two groups. There was a group of reports that really focused on regression, I think as it's thought about in autism, largely focused on language. And these are these studies here. So dating back to 2010, I'm sure I missed one earlier. But they tend to be, these are studies across a range of ages that use metrics similar to ours, looking at regression as defined in the context of autism spectrum or in an undefined sense, just by parent report. And they see, maybe between 15 and 35, ours being maybe a little higher than average estimate of the individuals having regression. And they are, some our patients are older, but many are in toddlerhood. There's a different group of papers that doesn't actually necessarily use the term regression as commonly, but sometimes do, and then you start to wonder, is this some of what we're capturing in older patients? And these are reports of individual patients, generally all adolescents and older, who are reported in the psychiatry literature who are having changes in their ability and skills if you read the papers. But the diagnosis that they're receiving are behavioral and mood diagnoses, sometimes with symptoms of disordered thinking. And so these, again, date back to 2010, some very small. The paper by Egger was particularly helpful in that they reported 7 adults, all of which had psychiatric symptoms, and they reviewed many of these other papers. I'm going to, a moment, spend a time on this report by the group Serret and colleagues in France, because I think they had some really nice data showing something that I think is really interesting here. So obviously this is a topic that's very concerning, I'm sure of interest. And one of the things I think that's most interesting about it is that in this group of older patients, many of their stories include the improvement and recovery of their symptoms, and people starting to propose which intervention was or was not taken may have been more or less helpful. So to me, this is one of the most exciting things I've come across in the field of Phelan McDermid syndrome in a while, because there are so many stories of improvement of symptoms in a fairly dramatic way in a domain where classically, from my training, if you hear about regression, that's not something that people recover from. So I think it's important to recognize the spectrum of what's being reported, and that not all these trajectories are only a reversal, but patients regain and recover, and it may correlate with some of the interventions that have been taken. So this is not systematic data. I think it's something that would be interesting to discuss and to do formal studies on. But many of the people who have reported these patients have noted that they have symptoms that can look like bipolar disorder or sometimes depression. And some of the patients are reported to respond to medications in the class of mood stabilizers, which is a broad group of medications, but there's definitely some that are mentioned recurrently in the literature. This is the figure I wanted to share with you from the paper by Serret, because it was one of the few where there had been systematic assessment of the same individuals both before, during, and after the period of their life where they had been assessed to have regression or the psychiatric symptoms that correlated with regression. So this was the Vineland scales looking at domains in communication and daily living skills and socialization. And in these 2 unrelated adults, there's a relatively clear pattern that they had their baseline function at the time of their diagnosis with Phelan McDermid syndrome. They had a continuing sort of at that level. There was a period in their lives in mid-late adolescence or early adulthood where that changed. Sometimes associated with other symptoms like urinary incontinence, which is not something that you usually think about in the childhood autism regression. And then these authors were highlighting that both patients had improved with a medication called lithium, but I don't want to overly emphasize the role of any one medication at this time in this talk. So in summary, I think the neurodevelopmental disorders can have distinctive patterns of development, and Rett syndrome is known for this. Historically, I don't think that Phelan McDermid has been in that group, but increasingly I think that maybe it should. I think it does have its own characteristic patterns of development and developmental trajectory. And it includes a phenomenon of regression that can be later and in some cases appears to reverse. And that raises a lot of open questions. So I think I'll mention them briefly, but I don't want to steal other people's questions or discussion. But these are the things that are high on my mind. Is what are the different? Is this a continuum, or is there really a difference between these 2 what appear to be slightly different presentations of regression? And what are the potential causes? Many things have been raised. And if there are really different subtypes of regression, and some of them appear to be responding to treatment, how important it is to be precise about that and think about how we can guide our interventions based on these specific, more nuanced conceptions of regression. So I think I'll stop there and we'll transition.
Thank you so much, Dr. Bernstein. Gold star, finished on time. We are going to put up our question. The question is up for poll everywhere, so please go ahead and get that answered and start your discussions. Thank you. Okay, everybody, we have about 5 more minutes of this discussion. The poll everywhere is up and ready to accept any questions that you have, so please submit them. And please feel free to vote on any questions that might be pertinent to you so we can make sure that our panel can answer them for you. Thank you. Okay, in about 2 minutes, we're going to start our panel. So if we could call the doctors and researchers up now.
People ready to wrap up and transition to the panel? Even if you're not ready to wrap up and transition to the panel, I think we're going to do so. Listen up. Shh. Okay. I'm Tazy Kohlenberg. I'm a child psychiatrist and also the parent of a teenager with Phelan McDermid who's had neuropsychiatric difficulties and regression. I'm incredibly grateful for this conference and for the time that all of these smart people are giving us. You've already heard who John Burstein is. He's back. And next to him is Ann Neumeyer, who is a child neurologist from Boston. She's an assistant professor of neurology at Harvard Medical School and director of the Lurie Center for Autism. Last year, she co-founded the first Phelan McDermid Clinic in Massachusetts. She works with the Autism Speaks Autism Treatment Network, and she's very interested in better clinical care. I think she's probably got a bunch of patients in this room who are glad to see her here. And next to Anne is Alex Kolivson, who's a child med. Who apparently needs no introduction. So at this point, we get to start with questions, yes? Okay. So the first question is, does deletion size impact the level of regression? Who would like to take a shot at that?
That was overwhelming. Thank you. I'm sorry that I couldn't be here yesterday. I heard Joe did a wonderful job representing. I'm glad I wasn't here, maybe because I may have not have gotten such an enthusiastic applause. So right, the question is, broadly, how do we figure out course of illness for kids? And in particular, and relevant to this conversation, how do we figure out which kids may be susceptible to having a regression? The answer, sadly, is we don't know who's vulnerable to a regression. We know for sure that if you have only a point mutation in your Shank III gene, that is adequate to have a regression. It may even be that kids with only point mutations are at higher risk, because it does seem as if, from sort of emerging data, that the prevalence of regression within kids who have just point mutations may be higher than the prevalence across the entire PMS population. And that is sort of the answer. This is something that we are extremely interested in. The challenge, of course, is that in order to do really good studies about the relationship between the type of chromosomal rearrangement, the type of mutation even there are many different variants, as you heard this morning, that may have a role. We need to have many samples in order to have convincing data. And that's why you guys are the most amazing and have been so unbelievably helpful in getting us to collect more and more data. So the things like the registry are really important. Participating in the natural history studies are really important. So we are actively working on that and really focused on trying to figure it out.
Anybody want to add to that? Okay, so the next question is, are there any methods to prevent regression? Just a small question.
I don't think at this time I don't think we understand the dynamics of what's happening well enough to think that we could forecast how we would prevent it. I think that there are some leads in other people's experience in terms of how we may deal with it, and potentially those in turn, one could see how those would really lead to strategies for trying to minimize the risk. But I think the first question we're thinking about is seeing if there are subcategories of regression, and then trying to better understand what children who have those different types are responding to and what they're not responding to.
I'm going to just tap. Yeah. So I'm just going to add also that ensuring that your child with Phelan McDermid syndrome has as good health as possible, and that's also in following preventative steps, making sure their thyroids are checked, and that they don't have other ongoing infections is also very important to helping them so that they might not have a regression due to another medical cause.
I think being very sensitive to it is critical. Often doctors will dismiss your concerns, right? Because they'll say, "Oh, this is just part of the syndrome. There are always these ups and downs." We're working to try to figure out some clear criteria so that this is taken more seriously. So I think, as always, intervening early and aggressively when you see symptoms emerging that change, that are different than what you understand your child to typically be like, we want to be very clued into that.
Yeah, I agree with that. I think that, to me, that's sort of mitigating and responding and recognizing things as quickly as possible. And I think that's something that's going to be happening a lot more based on the visibility that this aspect of the condition now has, and even just sessions like this.
And we will be making an announcement at the end of the panel that bears on that question. The next one is a fascinating question. Is there a point in a patient's life that it should be considered degenerative as opposed to a regression?
I'm not speaking from personal experience. I'm relying on the literature on these phenomena, and there's very, very few reports that are strictly degenerative. There are a couple out there within the papers I cited a few minutes ago, but in terms of the classic picture of a degenerative condition that leaves someone without any skills or potential for recovering, that was not a common storyline.
This is, I think, the same question coming again. What is the correlation size of deletion and regression? I think we've answered that. We'll move on. Any ideas about causes or correlations of various kinds with regression?
I can go. You want to go first?
So causes or correlations with various kinds of regression. I think that the literature seems to suggest that there are some environmental influences, in the sense that some reports have occurred when children move, for example, when they switch environments, when stressful things happen in the family. I think that the onset of epilepsy or an increase in the severity of epilepsy can be associated with regression. We know now, and this isn't as reported as much in the literature, but certainly by anecdote, that infection, changes in what we might think to be sort of their inflammatory state can induce regressions. So those are all possible things and all things that we're looking at.
Do you want to say something more?
I think that covered most. I think that the way the question was posed was very good. It's sort of causes or correlations. I think that we're left with these things being in either category right now, and with time we should understand better.
So the next one is there a correlation or a relationship between acquiring a new skill and losing a previous one?
That's the big question. I can't answer that. Can you?
So often we see that, for sure, where kids will make gains in one area and then often lose in other areas. And I don't have an explanation for it. But even with language, kids will learn new words and start using them with some consistency and then perhaps lose old words. I don't know that I would call that necessarily regression, but there's definitely a lot of shifting that goes on. And parents talk a lot about how long it takes to learn something and how much behavioral intervention often goes into learning a new skill. But then you're not reinforcing at the same time necessarily all of the old skills, and they can potentially fall off. So it's very challenging, because obviously this protein plays such an important role in learning and memory. And so it's you can kind of imagine why it's hard to hold on to as much information.
And I'd like to mention Barry Brazelton's observation in his Touchpoints work of the fact that a lot of energy of the organism, even in typical development, goes into new skills, and you can see disorganization, particularly in sleep, as people get ready to learn new things. Now, that's not at all specific to Phelan McDermid, but it may be true of our kids. Let's see. Is regression more common in higher functioning individuals with Phelan McDermid?
We've been discussing this leading up to the meeting, and as Alex was, there's some anecdotal evidence that perhaps it's slightly more common. It does seem that it's more readily appreciable. So, children who are functioning at a higher level on these developmental scales, it's maybe more obvious to people around them that there's been a change.
Anyone else? Okay, so how often is regression correlated with immune challenges such as illness or vaccines?
I don't think there's any systematic data on that question. I think of the multiple, they're really just ideas that have been put out about what's going on in the children who are having regression involvement of the immune system is an interesting and open question, but we don't have numbers or data that would allow anyone to say with any certainty that there are cause and effect relationships in that domain.
Yeah, and I think just because I heard the word vaccine in there, and I definitely would I would say strongly that we still want to have our children vaccinated by all means. Let's not go down that road, please.
Okay. Please elaborate on theories of why regression occurs in Phelan McDermid. Another small question.
I think the question might be, please speculate on mechanisms, but other than sort of naming some of the areas that people are considering, I think it's very speculative to try and lay out a narrative of if event A leads to event B, and that's why we see event C. Broadly speaking, people have thought about the biology of Shank III and the development of the nervous system and what's different about the nervous system. People have talked about the potential role of Shank III actually directly in the immune system. Some authors have proposed that actually the structural anatomy of the brain is related to the liability for the mood symptoms. There's been discussion of the role of the environment in combination with these, but it would be, to me, it would be premature and not entirely appropriate to try and be more precise or specific than that at this time.
Any comments on what genes might be associated with regression?
Well, I would give Shank III.
Yeah, and in fact, because we're seeing perhaps the prevalence of regression as more common in kids with just point mutations, I think we can isolate Shank III as the main gene responsible.
This one is an observation masquerading as a question. Why is retraining a regressed skill or motor function or word so much more difficult than it was initially?
So I think it depends a lot on the child's age, in the sense that if a learned skill like walking developed at 2 or 3 or 4 years old and then that was lost, just by nature of development, the brain is less plastic 10 or 15 years later, and so it may take more time. I think that's probably true of a lot of the skills.
Yeah, I sort of provide a comment and then maybe a question back to the other panelists. Is, to me, there may be a difference between trying to regain a skill that's been lost in the context of anxiety and mood instability and or depression, versus one that's lost in a toddlerhood regression or in the context outside of Phelan McDermid syndrome where it is a degenerative condition. So what makes it easier and harder could be different in those contexts. It may not be a monolithic regression that reacts in the same way or has the same dynamics.
Okay, thank you. Comments, the question is just seizures? Relationship thereof, please.
So we know that seizures, children who are having uncontrollable seizures have regressions. They're not as alert, they lose skills, and then when the seizures are treated, then they regain many of those skills and they become more alert and they can learn new skills. So very simply, yes, seizures can impact regressions.
My impression is that there are obvious cases where children have had severe seizures and uncontrollable seizures, and this impacts their function, and that is a large part of what's going on in those cases. There are also numerous reports where people have highlighted, gone to lengths to say that this child had regression, and there are no seizures. There is nothing different about the EEG. So there are likely multiple paths to things that can be categorized as regression. So again, that's my interest in being more precise about this and thinking about subtypes potentially of regression.
I guess I was going to say, just back to this question of the cause, right? I mean, regression is, as we're defining it, it's a loss of skills. There are many, many, many different kinds of regression. There's not going to be 1 or 2 or 3 or 5 causes. There's going to be lots and lots of causes: toxic causes, metabolic causes, infectious causes, inflammatory causes, epileptic causes. I mean, I think that on top of a brain of a child or an adult with Phelan McDermid syndrome, they're going to be more vulnerable by nature of the fact that that brain is not organized in the same way. And it's going to manifest with a whole array of clinical symptoms, including perhaps a loss of skills in some subset. The challenge is to figure out, how do we identify it early? How do we predict who's most vulnerable? And how do we figure out what the best interventions are to try to either, diminish the extent of the regression or even rescue the skills lost? John made a really important point. I feel like we're all frustrated because we don't have good answers to any of these questions. You're all frustrated because this is an extremely scary potential outcome. But yet, I think once the regressions have been identified, at least from the literature to date, there seems to be some emerging evidence that medicines and therapies can actually help, which is very good to know. So we can get kids back to where they were.
And that's a pretty unusual thing, broadly across medicine. There's not a lot of conditions where people are using the word regression and talking about it reversing and going away.
And I want to echo what John said also about the importance of you as parents advocating for your children when you potentially see a regression, and potentially even educating your doctor or nurse about that.
Okay. Question is, how common is regression in adulthood?
I don't know if I came across numbers that were broken down by adults versus children, and also the registry may be a better way to approach that. All of these papers that I looked at were, sometimes these convenience samples, so people who happen to be available, or patients that were selected on the basis of having had regression. So in terms of making the comparison about how many patients have it versus don't in an unselected group, that information is not as readily available.
The other important thing to know is that a lot of older adults who might have Phelan McDermid don't know it because they didn't have the testing done when they were children. And so there are many people here even who have only recently discovered that their adolescents or young adults have the syndrome. And so we just don't have that information yet. And maybe that's a question we can answer in 5 years better.
So that's just a great opportunity to plug the adult studies that we've been starting. The Phelan McDermid Syndrome Foundation actually recognizes this as a major priority and has funded our existing natural history consortium to expand to adults beyond 21. Right now that consortium focuses on kids from 3 to 21, but we now have additional funding to recruit adults over 21, and one of the key questions is exactly this: what's the natural history of adulthood, and what's the prevalence of things like regression among adults? And we've added new measures to try to capture some of those symptoms. The other kind of, well, 2 other points. One, as a teaser, we're now resubmitting our application for another natural history study to continue the ongoing study, which will obviously continue the work that we're doing, but one of the main changes is going to be to include younger children, so below 3, and older adults above 21. So these are things that we're very, very focused on. We also have adult studies going on, additional adult studies.
So that's it for questions for now, though you're all invited to descend upon these three people when we break. But I did want to, this is also a chance to announce something good, which is, excuse me, making an announcement. So after a year's work with enormous support from Alex and Anne's participation and the participation of some other experts in the room, we are getting started something called the PMS Neuroconsultation Group, ECHO. And that is a service that we will be providing. Jimmy Holder, part of our team, will be providing video case consultation to the doctors caring for people with PMS in remote areas who may not have confidence or experience. For people with PMS who have complex neuropsychiatric difficulties and or regression. So this is not seizure management, this is not GI, it's this thing that we're talking about today. And this will start this fall. It's a collaboration between the Foundation and the Seaver Center for Autism at Mount Sinai. All of the experts involved are donating their time to help patients and to encourage the growth of expertise around the country and potentially around the world in managing these issues. Details about how to access this will be available soon, both through the Foundation and through the Facebook pages. And so there's 2 slides of the kind and generous people who have helped make this happen. You might recognize some names on this one. And here's a couple of other familiar names. And I can't, as a parent of an affected child, I can't say enough thanks to all of the people who are doing this work. It's on top of enormous clinical loads, academic responsibilities, administrative responsibilities, research responsibilities, and it takes a lot of heart. So thank you all.
Sorry, I can't let that end without acknowledging that actually Tacey was absolutely driving that bus. We started having these conversations a year ago. But there's no way this would have gotten done without her.
Okay, we're getting near the end, but we have about 15 more minutes left. And while I make some announcements, I'd like for Dr. Craig Powell to make his way up to the front. Craig, are you around somewhere? Okay, so I've been asked to make all these announcements. First, we have a ton of research opportunities going on at the meeting here. The first is the registry help desk. It's right down the hall that way. Stop by, drop off your genetic reports, chit-chat with the registry team. They'll be happy to meet you and answer any questions you have about participating in the registry. The poop study is just right outside these doors along this wall. Go chat with those folks. They'll be happy to enroll you, and you can take a kit, I believe, and talk to them. I think you can take a kit home and ship poop back. Or if your kid is here, you can poop on site. Okay. There's also a team that's taking sort of 360-degree photos of our kids to look at sort of specific features in PMS. They are located just outside these doors and across the hallway. They're very close. You'll see they're set up right across. And then there are some other studies that are recruiting, and if you're interested in learning about opportunities to participate in research, you can go to the registry help desk, and they've got some flyers there. So, today's genetics talk was great, but it was just sort of an introductory talk on genetics, right? And so tomorrow we'll have an advanced genetics session in Heritage 2. That's at 9 o'clock. And then following that, there's going to be a little coffee break. And then following that, there will be genotype, phenotype findings and special genetic findings that will be in Heritage 4. So please come to those sessions if you want to deepen your knowledge of genetics. This afternoon, we are going to have a scientific symposium. It will be held in Heritage 2 and 3. This is really a researcher symposium for scientists to communicate with each other, share some of their findings, and talk about sort of precision medicine approaches to developing therapeutics in PMS. And families are certainly invited to attend that. There's, I think, fairly limited seating in that room. So, lunch announcement. Box lunch tickets are being sold at the Mercantile. You can pick the tickets up there and then pick up your box lunches in the atrium. Researchers and scientists, remember today we're having a luncheon, and that will be in Heritage 1. For those of you who haven't downloaded the conference app, you should do that. And then you'll want to accept push notifications so you can receive announcements as they happen. In fact, we have a new announcement, and that is we've added a new session. It's going to be on GI. It's sort of a GI meetup, and that's going to be at 5:30 p.m. today in the Bennett boardroom. At the moment, I don't think that's on the agenda, so just make a mental note of that and be sure to get the push notifications on your phone. Did I do that right, Nick? Okay, thanks. And then tonight, there's a dance party that's from 7 to 10 p.m., and the silent auction will end at 10. And you're supposed to wear your boots and your cowboy hat. Okay. I will. I'm going to wear my boots. Okay, toileting appointments. If you have a one-to-one toileting consultation, please make sure you keep your appointment. They are in the Nash conference room, and the schedule is posted on the door. Okay, Craig, can you come on up here? So this is Craig Powell. Many of you know him because he's participated in our other conferences in the past. He's currently a neurologist at the University of Texas Southwestern here in Dallas. He was recently named the chair of the Department of Neurobiology at the University of Alabama at Birmingham School of Medicine, and he will also be the director of the Civitan International Research Center, where he will lead research on developmental disabilities. Dr. Powell is just going to really just offer a few last reflections on PMS research.
Thanks, Geraldine. So I'm supposed to summarize a whole day of exciting and boring science. And I know you guys have... all I see in this room every day, every time I come to this meeting, is 100% of blood, sweat, and tears, and heart, and drive. Everybody in this room has family to deal with, children to take care of, work to go to, health to maintain of their own, hopefully, if there's any time in between, and everything else in your lives. And yet there are a couple of people in this room, and more than a couple actually, that take care of all those things and do 1,000 more things. And I just want to start by saying that two of those people, only two, are Geraldine Bliss and Abby Laganoff. You all, I know that there are changes in the board, and there's changes in the officers of this foundation, but I know that you all know the people that I know and interact with on a regular basis. People like Sue Lomas, people like Megan O'Boyle, who has run the registry as a non-scientist for many years, the entire board, and all the volunteers that make this meeting possible. And that's really all of you in this room, right? So I have personally contributed the money for the salaries of everyone that I've just mentioned, that they get for doing all this. And so if you think the thermostat in the room is a little bit off, or the weather outside isn't perfect, or your hotel lobby isn't as accessible as you might like it to be, or the room is a little too crowded to get in and out, all that's true, right? But really, they've worked enormously hard, hundreds of emails, extra trips to the hell that is Dallas, Texas. I can say that because I live here, and I love it, and it only snows and ices one day a year. But think about all the audio-visual coordination, the schedules, the changing the schedule on the fly. Think about all that and who's doing that, and it's a lot of those people I just mentioned, plus a lot more. There are about, I don't know how many scientists are here, clinicians and pharmaceutical company representatives, foundation board members, and others. There's at least 25 plus scientists that have been in and out of this meeting and will be in and out of this meeting during the course of the several days that is the conference. None of us are paid by the foundation to be here, although some people's travel is reimbursed to get here, right, to be here. And, you know, all of you are here, and just by being here, you're contributing to the research that is done, okay? I was talking to some of the adult parents up in the front yesterday, and, you know, just by the fact that your child has been diagnosed with a genetic change, that's a genetic difference, one of many, has led to the discovery that the Shank III gene is the critical gene for most of what we call Phelan McDermid syndrome. All of what we call Phelan McDermid syndrome. And then, you know, there are many other disorders out there, many other genes that are involved in other disorders that are similar or very different than Phelan McDermid syndrome. And everybody in this room at some level has contributed to this effort of discovery, and that is science, right? So you are the scientists as much as we are, and you're contributing as much or more than we are. And you guys as a foundation raise, you know, a few $100,000 a year, maybe more. I don't know the exact numbers. And I can remember this foundation, you know, when I first became involved or, you know, became aware of this foundation, there were a couple of dads at a table at the Society for Neuroscience meeting. Randy Riddle, I believe, yeah. And all they wanted to do was for someone to make a knockout mouse model of Shank III gene, right? And that was one of the goals, and to bring families and their kids together so that they could learn from one another and so that they had a place to be. Well, you guys have accomplished so much more than that in just a very short period of time, right? So your couple of 300, 400, 500, whatever, thousand, hundreds of thousands of dollars you raise every year has turned into literally millions of dollars in federally funded research, pharmaceutical funded research, foundation funded research, Autism Science Foundation funds Shank III research, Autism Speaks, Simon's Foundation. You guys are the main engine, the drivers of making that happen. Not just by your very coming to this meeting or existing on the web or being a Facebook group or being in the registry, but by the blood, sweat, and tears that you and every member of this foundation puts in every day for the year round. And so what does that $1 million in research turn into? What have all your efforts, all you've got is a panel that often says, we don't know, 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 we don't know. And a bunch of scientists who put up EEGs and squiggles and things you don't understand, and who cares? I just want my kid to be a little better, just a tiny bit better. Well, we've heard about at no less than 5 clinical trials of new drug therapies directed specifically at this population, Phelan McDermid syndrome. And that is something that 10 years ago I would have said there's absolutely no way I would be standing up here and talking about 10 years from now, right? Just unheard of, believe it or not. 2 of them are completed, more are enrolling, and even more are planned or just in the beginning stages. And there's more studies, as you saw over the course of the last day, really, being done on behavioral issues in Phelan McDermid syndrome, GI issues specifically in Phelan McDermid syndrome, poop collection, genetic studies that are still ongoing through the registry. People are still curating those data and making sure that the diagnosis of Phelan McDermid is accurate in every single patient, because that's required for the next step to figure out what percentage of your kids have X, Y, and Z complications, okay? Regression, for example. We just heard a great example. Epigenetics, whatever that means, right? Doesn't matter. These are important things that are being studied every day. So I think the message is that hope, which all of you have in spades and should continue to have, money, which you have or will be hopefully contributing to raising, science, which we love to do and which more and more people are becoming involved in, specifically with Shank III and Phelan McDermid syndrome, specifically because of you in this room and the people who are advocating on your behalf for free, unpaid, and family participation. All those things added together have led to potential new therapies and more hope than ever for Phelan McDermid syndrome, okay? I mean, we've heard about drugs, IGF-1, oxytocin, ERK-MAP kinase inhibitors, new molecular targets, new targets for epilepsy that are being targeted in the Shank III population specifically. Behavioral plans, how to fix aggression, how to fix tantrums, how to get evaluated. I learned a new phrase, FBA, functional behavioral analysis. It's different, apparently, than applied behavioral analysis, although some of the same principles can be used for both. Communication strategies. Many of you have the luxury of being able to use pads or iPads or communication cards or just cards or gestures or grunts and moans and other signals. Clapping is a big one that we've heard about at our table yesterday. And then gene therapies we've heard about. The possibility that maybe we could fix the underlying genetic problem. And those studies are being thought about and ongoing in mouse models, ongoing in human neurons in a dish. And so this is the hope, right? The best example I can give you is who's heard of spinal muscular atrophy? Keep your hand up. Who's heard of ALS, Lou Gehrig's disease? Okay. Who's heard of a baby getting Lou Gehrig's disease, or essentially that, right? A few people, scientists mostly. But the fact of the people at my table from yesterday. So these are babies that are born with SMA, spinal muscular atrophy, and they basically have infantile onset Lou Gehrig's disease, and many of them are no longer with us after age 5. Not all of them. I'm just don't quote me on that, the exact age at which there are different flavors and different varieties. But these kids progressively lose motor function until they can no longer control their diaphragm or their swallowing, and they pass away uniformly before age 10 or 12, okay, in certain forms. There's now gene therapy that's been done and is FDA approved and costs a lot of money that goes into the spinal fluid that turns on another gene that looks like the gene that's missing and changes it, and these kids are now living beyond age 5 and they're walking, whereas they were essentially what we call just a floppy baby that's never going to learn to walk. And that's a miracle, right? No one up here is promising a miracle for Phelan McDermid syndrome, but we're working as hard as we can to get there. And so there is hope. And you guys are the catalysts and the drivers of all of these new discoveries, each and every one of you. It's not just your board. It's not just this mysterious website and this mysterious registry that you put stuff in and you feel like you're getting nothing out of. You're not. That's not the case at all. So the foundation isn't some magical entity that provides all this and makes this happen from nothing, right? It's you. It's your families. It's your friends. It's your communities, your social networks that drive this, right? You now have an executive director who makes a salary, who's in charge of making everything work the way it's supposed to. You have a registry that costs money to keep it alive, It's not just you put it on the web and you enter your data and it just happens and it stays there magically. There's a cost to keep that going every year. There's now a coordinator for the registry, and the foundation pays that person's salary to curate the data. You need desperately to have a scientist in charge of helping your organization decide what's the best strategy going forward to use your resources, the families, the registry, the gosh knows what else is coming on the horizon, to make the hope a little bit more promising, right? To nudge things in the right direction. And all that takes money. I think you need a shake-up on your scientific advisory council, right? So a lot of us have been are directly involved in Phelan McDermid syndrome research and have just by hanging around have ultimately benefited from some of the dollars that go to researchers directly. And that's helped to fund UT Southwestern, for example, with a little bit of money to do patient sign-ups for the Developmental Synaptopathies Consortium, and Stanford for the same reason, so that you don't have to travel quite as far, many of you, to get to one of those sites, okay? And then NIH adds to that hundreds and hundreds of $1,000s every year, and it's never enough. So you are the only ones who can go out and raise money. I don't personally benefit from that money, so I feel like I can feel comfortable saying that if you want more of this science to be done, you have to go out and raise money for the foundation, not for me, not for the scientists, not to put money in anybody's pocket, except to keep this organization alive and to keep the hope coming and to leverage these relatively small dollars into these millions of dollars, into these studies, into the letters of support that go to multiple grants that are written by Geraldine and Megan O'Boyle, and they have to check out everybody's IRB, and there's a lot of work that goes into all that. And they're not going to tell you that. They're not going to complain about it to you, because you guys are all you're all families and you're all friends and colleagues, right? So there's more than 10 animal models, different animal models of Shank III gene mutations for Phelan McDermid. Rats, mice, you name it. There's more than 10 of you in the foundation, way more than 10, that have donated cells, but there's more than 10 independently created human neurons in a dish from people with Phelan McDermid syndrome. Drug companies are screening drugs on those neurons and in those mouse models. And none of it would have happened without this foundation. None of the science would have happened without this foundation. None of the registry would have happened without this foundation. And so you are a part, each and every one of you is an integral part of this research. And you may get an email because you're part of this foundation, or a bulletin in the newsletter that says, if you live near Mount Sinai, they're doing clinical trial X, Y, and Z. If you live at near Stanford, they're doing this. If you live in Dallas or Birmingham or wherever around Chicago, you name it, participate in this study. And that's a very personal decision, and you have to think hard and long about what you want to do and how you want to contribute personally. So this is not for you to raise money for scientists or me. It's money to lobby Congress, to lobby NIH for increased research dollars focused on your disorder. It's money to lobby the pharma industry to stay engaged in this particular rare disease. It's money to maintain, curate, and grow the registry, to develop standards and clinical centers of excellence, right? And the ECHO project, where there are experts that are virtual to whom your doctors can speak. So the science you saw today and yesterday is all about you. It's all for you, but you're an integral part of it. And so I would just urge, I think you all deserve a round of applause for being involved, for coming to the meeting, for bringing your kids, for not bringing your kids. Thank you to those of you who didn't, because there's a good reason you didn't, and thank goodness, right, for you and for everybody. And so I'm incredibly grateful to everybody for being here and listening to sometimes boring talks and sometimes really important pearls of wisdom. Functional behavioral analysis, GI transit studies, don't do this diet, do that diet, watch what happens when you change this or that. And all the scientists are available to hear your stories during this meeting, and I just want you all to realize how important what you're doing and what your foundation is doing. And I don't represent you or your foundation. I'm just here as a scientist trying to make a small difference in the scope of things. So give yourselves a round of applause. And when you leave, take your hope and your frustrations and raise money for the foundation, because it doesn't go into my pocket, and it certainly doesn't go into the board members' pockets or anybody, any of the volunteers' pockets. Thank you.
Amen. Craig Powell. As Craig was talking, I'm getting text messages from board members saying, wow, I mean, Craig is quite the pitch man, isn't it? He hit a home run there. You could not have said it better. I know that's exactly what Sue Lomas wants to convey, and all of our board members as well. And I didn't expect that, Craig. I thought you were going to talk science, so thank you. Okay, childcare ends very soon, so you guys are going to need to head out to pick up your children. But I just want to say a final thank you to all of our participants, to our speakers and our panelists, our table discussion leaders. Thank you guys. You've done such an important job. Our note takers, our AV team. They've done a terrific job. Thanks, guys. Julie West, our Poll Everywhere czar. And most importantly, for you, the parents and caregivers, for contributing your knowledge and your experience to the Phelan McPosium. Please enjoy the rest of the conference.
So, as our children are older, and you guys didn't address it, the fact that regression and degeneration